Greetings and welcome to another fantastic episode where we get to dive into SingularityNET and the ASI Alliance's deep tech, deep research, and development along our AGI, beneficial AGI goals. I am so honored to be joined here today by the amazing team from Rejuve.bio. First, we've got Kennedy Schaal, CEO of Rejuve.bio, Mike Duncan, Chief Science Officer, Hedra Yusuf, Chief Technology Officer, and Abdulrahman Omar, Lead Machine Learning Engineer. It's an amazing crew. I really find fascinating and the complexity of the team that Rejuve.bio has built. Medical, longevity, machine learning, AGI developers. It's an amazing mix and a very complex team. Perhaps, Kennedy, you could start and kick us off. Tell us a little bit about Rejuve.bio, what you're aiming for, your approach, and maybe dive just a bit into the complex team needed to achieve this mission.
Well, thanks for having us, Haley. Rejuve.bio aims to solve one of the biggest pain points in biomedical research, and that is translational medicine. There is something that's often referred to in research as the translational valley of death. And what that refers to is getting literally from the lab bench to the bedside. And that is the point at which most medicine and most research fails. And that is an area of biomedical research that we see where AGI and AI can help the most. Specifically, the platform that we're building goes beyond the generative AI tools that have started cropping up in drug discovery recently. We are putting together a suite of algorithms that all work together, functioning much more like a brain, like we want AGI to function, which, as you can imagine, in biomedical research makes a lot of sense because sometimes you're dealing with data sets that maybe aren't as complete as you want them to be. Maybe they're small, maybe they're too large. Maybe you have N of 1 studies, maybe you have a couple of studies. Eventually, you might have synthetic data. So in biomedical research, you have this huge diversity of data sets, and it will take more than just generative AI to solve for these pain points and get to a point where we can model systems that much more closely approximate human biological systems to hypothesize what might actually work in a human system. Right now, the normal drug discovery pipeline is that you find a small molecule, you test that in a petri dish, what we refer to as in vitro testing. You approximate that in small animals, usually a mouse or a rat. You might get to a larger animal, like a non-human primate, and then you spend all of that money, all of that time, and then it might still fail in a human. What we hope to achieve with this platform specifically is to decrease that failure rate by using advanced artificial intelligence to get a better approximation, better hypotheses of what will actually work in a human system before all of that capital is spent on something that eventually doesn't even end up working. So that is the main problem that Rejuve.bio hopes to solve with our platform that we're building. And then specifically for aging related diseases, which is our main bread and butter, it can be said that 90% of chronic diseases are due to the aging process. We have been fortunate to live in a time where there have been advancements in medicine and human health, and as a result, we are living longer, but with living longer comes more diseases. And so, but also we want to extend that human health span, that point in life where you are healthy, of course. And so by looking at by amassing all of this data, we are hoping that we can use this Proto-AGI system to find patterns that weren't previously visible to the human eye, something that an advanced AI can identify and look for new aging pathways, or better solve for ones that have already been identified, and actually go after therapeutics for aging related diseases that can actually practically be developed.
That's fantastic. It's just amazing to think of all the complexity in biology. We, you know, learned it in fifth grade and such, and we think we got it then, right? But we've seen all the science coming out, and biology itself, the pace of understanding of how biological systems work, how the human system works, how aging works among animals is increased so much in the last years, and just accelerating, and how AI is driving that forward. But some of the complexities of developing these solutions haven't changed. They haven't been updated. And so we need something really intelligent, this really intelligent pipeline, to be able to make that AI applicable to solving these problems and not just understanding systems in a really complex way in our complex system. Thank you. So tell me a little bit about the team, everybody that you have working on this goal, and how that team has evolved over the past years to be able to address these complex translational solutions to health span and longevity.
Well, I'm extremely lucky. I have a ridiculously qualified team to be working on this goal, I believe. And it's I think that it's you know, somewhat an act of the universe or fate or whatever you want that's brought us all together at this time to solve this problem. It's um I myself, I've been a longevity biologist for 20 years now. I was trained by Michael Rose at UC Irvine, who's one of the fathers of the longevity field. And so this is a problem I've been working on. This is all I've known in my professional life. And I've had the privilege of working with Ben on these AI systems for almost as long. So it's a tool that I've used in my studies, using our model, the Methuselah fruit flies, which is our proprietary data set, super long-lived fruit fly that have been bred over 400 generations for 44 years. And so that's been a huge part of my life for about half my life now. And so that's where I've come into this thing. And then Mike Duncan, of course, is very qualified. He's a master of public health, applied mathematician, physician's assistant, just very, very qualified. And Hedra is a brilliant AI scientist, and she's always 10 steps ahead of me before I even ask, which is amazing. I don't even know how she does it. She's just a dream. And then Abdu is so brilliant and has acquired so much biology knowledge just from working on this platform that you would think that he has an advanced degree in biology. It is just like amazing. And what we've all built together is just I could not be more proud. And then our team members that we have that aren't on this call, Axel Schumacher, who's another pretty prominent aging biologist, and then Isahak, who did all of our UI UX for our platform so far, has put together some really brilliant visualizations of the hypothesis generator and our Atomspace visualization, which is not only crucial to our tech stack, but to the ecosystem's tech stack as a whole. And we have Vicky Yang, who's our product manager, who's done a really great job. And Of course, Lewis Farrell's done our marketing, and we just have a really, really great team. I just feel really lucky to that we've all been brought together at this time to drive this forward. So it's been really, really great.
It's so amazing how the development of AI can't happen in a vacuum, or perhaps shouldn't, or will be slowed down by just being developed for its own sake, and how these real world use cases that bring in real world complexity and answers that we all want really drive the synthesis of these complex teams that can then build the AI that build real solutions. Mike, I'd love to hear your perspective coming from more of a medical, more of a biology background, but then also the technical as well, on how the complexity of bio can be translated into AI, and the complexity of AI can be used to address these problems.
Well, something that I've learned about longevity biology and the work that I've been doing with Rejuve.bio and previously in Motzi AI, which is sort of the previous incarnation of this company, with Ben in Hong Kong, is that there's no one magic bullet to solving longevity or maximizing health span. It's we've had one of our key findings so far, working with the Methuselah flies, and previously with genomes of supercentenarians, people who've lived 105, 110 years old, is that the changes in the genome are widespread, and complicated. And this is true for like any kind of complex disease. I mean, I think that's become very clear in the genomic medicine age. And so that kind of, there are thousands of different changes in the genome that are contributing to this, to longevity, and like the human brain just can't encompass that. So I think, AI is key to finding, the understanding the root causes of aging, and for finding the solutions.
Yes, and none of those solutions are easy. What are some of the challenges that SingularityNET and previous incarnations have experienced in applying AI to the biological realm in longevity or otherwise?
I think a big problem has been in terms of, life is super complex, and there's the amount of data that's generated to understand it is vast, and has been done in a way that different teams have their own processes. Basically, you've got like a huge problem in the beginning. A huge problem was just like there's like many different names in the literature for a particular gene as they're being discovered, and so there's a huge amount of just incompatibility among the data. And then I think that's been changing, especially recently. I think because there's I think it's kind of an obvious problem, and I think there's a huge amount of resources are now being devoted to solve that, in terms of grants and stuff, to like make, there's sort of what's called fair research principles, where scientific work should be done in a way that the data is transparent and accessible for other people to use as necessary for science to advance as a whole. And I think that's much more accepted now, and I think much more, I think compared to when we started, even like approaching 10 years ago, like at least when I started doing this kind of work, there's like a whole generation of people who've been trained, biologists who've been trained to understand, data complexity and data science. And so I think there's a cultural moment that's been achieved, where a lot of the work now is being done in a way that is, under fair principles, so it can be, you know, usable by you know, on a global scale to advance medical research.
And that's so important that everybody can be involved. This impacts all of us. And this just natural tendency of humanity towards interoperability and working together, even across silos the best we can, is so hopeful, right? We're seeing this in the space of open source LLMs. We're seeing this in the space of crowdsourced biology and other sciences. People want to contribute and people want to make information available so that everyone can contribute and solve these hard problems together, which is the only way that we'll do it. Hedra, I'd love to touch a little bit on OpenCog Classic, the move to OpenCog Hyperon, and how important the Rejuve.bio type projects, these biology projects, have been in that move. From my understanding, one of the reasons that we understood we really needed to make a more scalable OpenCog Hyperon, first of all, we could see in the biospace that OpenCog Classic was able to generate some interesting results. There's promise here. We can see that this idea and this tech stack is working, but it's not scalable in its original format. And so we made the jump to Hyperon, which is still catching up to our use case here in Rejuve.bio. Can you talk a little bit about these transitions from OpenCog Classic, what we saw there, the limitations, and how that spurred movement into the Hyperon development?
Okay, thank you for having me, Kelly. So when we start working on Mozi projects previously, then that's when we try to apply OpenCog Classic to the biology projects that we have been working on. So one of the things we developed back then is the concept of Bioatomspace. So it's a unified Bioatomspace which contains different types of bio entities and their interconnection. And part of the reason the Bioatomspace is actually crucial to many developments is OpenCog being reasoning AI or reasoning-based AI system. It requires a knowledge graph to walk through the graph and come up with some sort of conclusion or do reasoning over the graph. So that's one key feature to have, and the Bioatomspace being a graph interconnecting different types of bioentities, genes, proteins, and their interaction. It was something that we were able to apply OpenCog on, and we find interesting results, including being able to query this huge graph over the query engine of back in OpenCog Classic, and being able to see the graph, which is a subgraph generated from user's query, and for the user to be able to see their interconnection and analyze results. And we were able to apply PLN or reasoning engine of OpenCog also, and then come up with some inferences which tells more about the what hidden patterns exist in the knowledge graph or in the Bioatomspace as well. So there were a couple of research that we have done back then, and moving back to the Hyperon system, basically the DAS would be something that would be crucial for the Bioatomspace, which means it will be distributed across different resources. Back then, due to the resource limitation and everything, we were only having the gene annotation system, which only annotates a gene, and we have a Bioatomspace for that. So now we are working on the platform, which is very vast and which considers as an input every bio entity that we can have in the Bioatomspace. So it's something that we brought back from the developments we have done before, and we expanded it even more to do more like the hypothesis generation service, which is backed by the Bioatomspace, and the query engine which we have at the moment, which will be a crucial development point for distributed atom space or for DAS, where we can distribute this huge billions of, in fact, trillions of nodes and edges connected to each other, which will be distributed across different resources and seamlessly for the user to be able to query them from the DAS endpoints through the Meta Hyperon interfaces would be something very, very crucial. So I'm glad the Bioatomspace, or what we have done before, is encouraging, or at least being the core point here for the development of the DAS and any other graph-based algorithms that are being developed in Hyperon. So that's something crucial about the Bioatomspace.
And it's so interesting to see the world of narrow AI, neural networks, LLMs catch up with this symbolic, that's a very sub-symbolic AI area, which is great at grasping some of the patterns and some of the connections, but the symbolic knowledge, which grounds it in reality and does away with hallucinations, which we just... we can't tolerate in biology. We need to have real factual information. Now LLMs are catching up to what Ben, the OpenCog team, and everyone have understood for a long time. We need to ground AI in real world knowledge. We need to ground it in graphs, in metagraphs, in the distributed atom space, and not just leave it up to the AI to make something that sounds good, right? Which can work for Twitter, but not so great for medicine. Seeing the world catch up to that, and how we're able to take our advanced graph knowledge and understanding and start to integrate it with LLMs and contribute back to that space has been exciting as well. And as you say, we need something as large, as complex, as comprehensive as the distributed atom space to be able to encompass just what humanity knows about biology. And we need something like Meta to be able to query an advanced graph like that. So by having these use cases, we're driving forward the development of these technologies and giving them something massively beneficial for everybody to work on. It's an amazing synergy.
Yes. With that, I would like to add maybe the benefit of explainable AI is actually coming up to a core point here, because with the current LLMs or AI in general, with a neuron approach, it's more of like it will give you the result, but you don't know how to explain it, or you don't know how it reaches to the conclusion. So with the neural-symbolic approach, with a reasoning approach, you can trace back how the AI come up with this conclusion, and the Bioatomspace is a core source in our reasoning engine, or reasoning for the hypothesis generation or any other things that we would do. As a source, it will provide you the guidance on where the knowledge comes from and how these things are interconnected to each other to reach to this conclusion. So the biologist is guided through the process on how this conclusion or this AI is coming up to this conclusion, or how this knowledge is being generated. So that's something crucial, and that's where the AI or this big tech companies are coming up to. So we're glad to be here and to work on this project.
Absolutely. On the leading edge of such a critically important for both the safety of medicine and also for regulation and for trustability, and building a system that we can show why and how it works, and maybe even derive further insights from that knowledge.
For sure.
Thank you, Hedra. And then Abdul, can you share a bit about how machine learning plays such a critical role in this piece, and all the components that you're helping to build out that we'll be demoing a little bit later today?
Yeah. So currently, as Mike said, biology is the most complex area in the sciences, right? We are here through millions and millions of years of evolution, so we can't just understand things out of like easily. So machine learning and the current AI helps us to take this vast amount of data and can understand patterns that exist so that we can connect certain mutations or certain genes to traits and phenotypes so we can target those areas. And the nice thing about here is this has an actual impact on humans, right? You can develop a drug, say, for a rare disease, and this will help some people who are suffering from this. Coming back to our platform, especially specifically the hypothesis generation platform. So what we are calling hypothesis here is a mechanistic plausible explanation that connects a mutation to a gene, and then through that gene to a biological pathways, and then a phenotype. So this is very crucial because, as Hedra mentioned, biology is one of the areas that you need to have an interpretable and unexplainable answer to your questions. So when ChatGPT or Claude AI gives you some answer, you really don't know how it got, which neurons fired and which neurons activated. But this kind of tools are useful by themselves in a lot of areas, but in a lab, you really need to know what's going on behind the scene to see what are the connections that are being activated. And in this aspect, the symbolic reasoning helps, and combining with a neural system like a large language model, it will give you the best of both worlds, where you have a predictive system and a reasoning system that's also explainable. And this is the quite attractive aspect of using Meta and Hyperon, because it's very expressive enough to allow you to do both neural programming and also symbolic reasoning, and that's one of the main benefits we have taken from this system. So our hypothesis generation connects, like I mentioned, a mutation to a phenotype through genes and pathways, and we do that by using a large language model to predict a list of potential causal genes, and then verify that through our probabilistic reasoning system to kind of select the most plausible hypothesis. So this, I believe, will help biologists in quickly testing prototypes so that they know what works and not what works before going to the lab doing experiments.
It's thank you so much. What a great description and explanation. The translational piece is so critical in everything that Rejuve.bio is describing and doing here. We need to translate what we know about biology, the research that's been done, into hypotheses that we can check. And we need to translate that into animal models. And we need to translate the results of animal models into other animal models, into human models, into more knowledge that can grow this system and synergistically feed advances in so many ways, from the research to the implementation to the benefit, and none of it's simple. It's all highly complex. Kennedy, can you speak just a bit to what differentiates Rejuve.bio's approach, how you would describe that, perhaps briefly, to other bio companies' approach to longevity, to biology using AI?
Well, I would describe the difference in, the core architecture that we're building in our platform. Obviously, going, as I said before, going beyond just generative AI, which is kind of in biotech right now is sort of a pioneering attitude to have. Biotech is becoming accepting just now of the generative tools that are cropping up. You're seeing those becoming more used in general, although people are still a little bit skeptical of them. Granted, once an AI developed drug does get approved and is on the market, I think that the floodgates will open and it will be good for all of us. But so far that hasn't happened, and I believe one of the barriers to entry that we are seeing, as Hedra was just talking about, a lot of these AI tools do not explain themselves. And that is a point of discomfort for a lot of researchers because, when you go to these regulatory bodies as a researcher and you're filing for an investigative new drug or you're filing to do your clinical trials, you have to submit a lot of paperwork that basically shows why you want to test your product or your new drug or whatever. And you can't just like cite, oh, I used AI. That regulatory body is going to be like, so what? So we're building into our system, that it gives you this nice explanation of how it generated that hypothesis, why it got to where it got to, why it got to the endpoint where it generated for you, so that you have this nice report to file with your paperwork for those regulatory bodies. And we're hoping that will make it much more research friendly to researchers, both in biotech and in academia. And then also the use of, as I said, multiple AI and ML algorithms working together, centered around the knowledge graph. Drug discovery, as I think everybody is gleaning from this conversation, is a highly complicated process. Like, you're not even, you can't even just like hook up an LLM to a knowledge graph and call it a day. Like, even just those two things are not going to get you there. Like, you're going to need, it's much more complicated than that. You need things to ground it in real world and truth. It needs to check itself. And so what you're finding in a lot of these generative AI tools that are being used by other companies and developed by other companies is they are making headway with finding targets that are succeeding, that are getting past the lab bench and into the clinic and into phase 1 and into phase 2. And there's a couple out there that are starting to do well, but they keep like kind of hitting these ceilings. And even so, a company will find 20 targets or whatever that look promising, and still maybe like 50% of them will make it to the next level. We anticipate that with our system, with using multiple algorithms, with using the Bioatomspace, that we'll be able to take that success rate beyond that 50%, like up higher to when we find those targets. And so that is the advantage that we see in our architecture versus just using a generative AI tool for drug discovery and translational medicine. So
that's incredible. Both the interpretability, which is of course a huge topic in AI. Special tools have been generated and released regularly just for interpretability of LLMs and their systems. We have this built in. This is a foundational, fundamental. We think not only do we need to understand what the AI is doing, it needs to understand what it's doing and how it's working. But this interoperability piece that you brought up is so important as well. There is not a wide recognition or understanding or interest in cognitive in architectures in AI. Generally, the neural networks aren't what I would define as a cognitive architecture. They're an approach. But when you start looking at an architecture, you look at multiple AIs interoperating, working together, coordinating an action to be able to answer these more complex problems. LLMs, neural networks, can be an important part of that cognitive architecture, as Abdul says, providing the hypothesis in natural language, understanding the request in natural language, being able to communicate is so important, but it is not sufficient for systems that can really dive deep into and generate new understanding, new hypotheses, new research, and see how those will work. And another component that SingularityNET and other AGI researchers are on the forefront of is this cognitive architecture, this interoperability of AI systems, and how do we build them together? Again, another super feature of the Meta platform, Meta language for platforms. So I'd love to dive into the Rejuve.bio Proto-AGI translational medicine platform. perhaps, Mike, you could give us an overview on what this means and what this looks like and its future outlook and intentions as well.
Sure, Haley. And what we mean by a translational medicine platform is a system that will allow clinicians to be able to work with their patients as in what's called precision medicine, or N=1 experiments, where each person is a unique system, and treating maximizing their lifespan is going to require many different interventions simultaneously. And I think that and so allowing a clinician to have access to experimental data and run experiments by comparing other patients, and working with other researchers in clinical trials as part of clinical practice is, I think, what's going to make this, I think is what's unique about our approach, and what's going to actually make all the amazing amount of research and discoveries that are going on in biomedicine actually available to everyone.
That's fantastic. Thank you. And perhaps, Hedra, I know we have some demos, but perhaps you could describe as well what we're going to look at today in this platform.
Sure. I will be able to present the annotation system where the biomedical researcher would be able to search what he or she wants to know more about any bioentity, and we provide. So this is one of the services in the platform, in the Rejuve.bio platform, and this is called the annotation service, where the user would be able to build their query or to be able to know more about the bioentity they are interested in. And this is the interface where you got all annotations you have done previously. And if you want to select a query template, which most biologists use, or frequently asked queries, or you can also build your own query. So one of the interesting thing in this work is we have a unified schema where you can tell all the components we have in the Bioatomspace in a form of this tab. Here you can click on it and you can select whatever component or whatever entity that you are interested in, and it will show you what other connections it has with other entities in the graph or in the Bioatomspace. So here I selected a gene, and it showed me everything a gene entity would be connected to inside the Bioatomspace, and I would be able to select any other entity that I want. In this case, it's a promoter, and I also add an enhancer or any other entity that you will be interested in, and you build the query in such attractive UI interface, and then you run the query. This will be very helpful for the user because they would be able to know which entity is related to which other entity. And since the unified Bioatomspace contains different types of databases across different data sources, they will be able to know which data is actually in here and which data is not. So there is also an option we are planning for a custom data to be uploaded so that their research or the things that they did in the lab can also be uploaded to this, and they can do more with what we have in the Bioatomspace and with their custom data they have in backend and in their labs. So in here there are some filtering options, and we have also an LLM explaining the graph in a natural language and showing how it would connect one entity into the other and what type of connections they have and other things. It can also have a few more things like showing what the frequent connections are and their numeric representation or the metadata. So it's a kind of exploring or searching something that you want to know into the bioatom space, where we have a unified bioatom space which contains different data sources. Yeah, this is the annotation service. And maybe Abdu would show you the hypothesis generation, where we do the neural-symbolic approach.
Wonderful. So right here in this demo that you've shown, we're looking at how can we dive into a gene and its related entities within a biological system and explore those relationships to maybe find relationships we weren't aware of, or to even analyze if a lab wants to do this for themselves, how their own data can fit into their own research, can fit into a broader picture that's been uploaded.
Correct.
How do we keep the bioatom space up to date and filled with the most relevant and current information that's being generated in research?
Yeah, that's an interesting question. Right now, what we are exploring, or what we have inside the bioatom space, is what is out there. And with the AI systems that we have, it's actually quite easier to kind of integrate whatever knowledge is out there and extract knowledge. Like we used to have, actually, previous to the LLMs, we used to have a traditional AI model which reads an article or a publication that's out, and then it goes through the research or the paper, and it extracts knowledge or a relationship between entities. It knows entities, biomedical entities, and then their relationship, and it imports those knowledge back to the atom space. So it's kind of enriching the bioatom space through this AI system, and there is an extension of that work in this platform as well that we are currently working on. So the latest publications or any other things would also be imported based on their area of interest, or it can also be uploaded by the user depending on their choice or what type of publications they are interested in. So there is an upload option for the user to be integrated. Most of them are actually for the LLM to be able to guide them through this annotation or any other things that they are being done, or this knowledge can also be extracted and saved back to the atom space for enriching it more.
That's amazing. Thank you very much. Yeah, Abdul, please, I'd love to have you share with us the demo of the hypothesis generator, another key component of the Rejuve Proto-AGI translational medicine platform.
Currently, in this example, what we have is a mutation on chromosome 16 with this is its DB SNP ID, RS142, and so on. And that trait, this mutation is known to be associated with obesity. And so usually, especially in the European population, where people who have this mutation from on their chromosome have higher BMI levels than those who don't have this mutation. So we are asking our hypothesis generation system, given these two inputs, which is the mutation the user is interested in and the phenotype it wanted to explain the connection, to generate a plausible hypothesis that's also interpretable and easy to understand. So that's what we are doing here. So the system goes through a list of processes, such as predicting the most likely causal gene. And then after predicting that gene, as you can see here, it will be validated as in, are there any supporting evidence for that gene to be the causal gene, such as are there any enhancers that this mutation is occurring in which this gene, which regulate this gene, or are there any transcription factors that bind to that area where this mutation is happening, and does this gene affect that, and so on. after validating the causal gene. So this is where the hypothesis goes through multiple potential genes that are that might be causally related to this mutation, and selects the one that has the most evidence. And then after that, we will try to see what other genes are co-expressed with this causal gene and find a co-expression network. And after that, it does a enrichment analysis where it goes through each biological process and pathways that might be altered by this gene, hence by this mutation. So this is how a higher overview of the system. Of course, there are a lot of work going on underneath, but as I said, it's like trying to get from a single point mutation on the DNA to a phenotype that the user is interested in, and essentially filling in the gap between these two and having a mechanistic explanation to show why this mutation is affecting this gene. So once the system is done going through potential hypothesis, it will pick the most likely and display to the user. So in this graph, you see here on the left side, sorry, there is the SNP or the mutation the user gave us. And on the right side, you have the phenotype. So all of this hypothesis, potential causal hypothesis, is generated by the system. And it's explaining how this mutation affects a causal gene called IRX3, which is known to be related with obesity and fat cell differentiation, and through which links there are other biological entities here like transcription factors, transcription factor binding site enhancers, and other genes which are near the enhancer. And then we have the co-expressed list of genes that are essentially forming a pattern. And now once we have this co-expressed list of genes, we want to do an enrichment analysis to get the most relevant processes or pathways that are affected by this mutation. And as you can see here, the top ones that are selected by the system seem all to be associated with obesity and BMI levels, such as adipose tissue development, fat cell differentiation, and so on. So the user can go through them and select whichever they want to look at in depth, which will give the explanation for that hypothesis. In addition, we have an LLM system that can take this graph and explain to the user what's going on here in a textual format, in a natural language format. And also the user can chat about this graph to understand more. So like you mentioned earlier, Haley, this is one way of grounding the large language model through so now this is grounded on this graph, this hypothesis that's generated from the Bioatomspace, and this will help users have more factual information than hallucinations, because it's grounded on an actual knowledge that exists in a knowledge graph. So this is the hypothesis generation system we have, and it's being used for research in-house.
That sounds amazing. That was actually my next question. Can the team speak a bit to how we're using this ourselves, what results we've seen or we're looking at, and how these tools are helping us right now?
I mean, our own biology work is centered on the Methuselah flies, which are a strain of fruit fly that has been experimentally evolved over 40 years now for longevity. And so now we're currently trying to take apart their genetics and figure out what is the systems biology that allows them to do this. And so we're approaching this problem is sort of guiding our our development of the platform, and through that the development of a particular use case for Hyperon. We're just in the beginning stages of that.
Kennedy, I'd love to hear your take on that, how we're using these systems right now and the results that we've seen from the research we've done.
Yeah, so as Mike said, we have done some research into the Methuselah flies, some cross-organism analysis. Interestingly, we found, as Mike touched on earlier, we found in some supercentenarian data, he had touched on the widespread mutations that we see in the genome of supercentenarians, and we found that the mutation burden in supercentenarians is about 20%, and in our analyses of the Methuselah flies, we found a very similar mutation burden, and so that is an interesting similarity that we found that we think points to some support to the flies as a good longevity model, and with the use of this platform in-house for the cross-organism analysis between flies and publicly available human data sets and other data sets that we're able to collect in-house, we'll be able to find some good pathways to explore for longevity therapeutics. To date, we've found a few transcription factors in the Methuselah flies that we think might contribute to their longevity. We haven't done quite enough research yet to fully say that we're certain, but results look good so far. So, but we hopefully we'll get to a point where we can use the platform we're developing to explore that further. But yeah, so in-house our flies will be used in conjunction with the platform we're developing, and the cross-organism analysis is the big advantage that we see in not only further supporting the Methuselah fly model as a viable model for longevity, and like a good economic model, because they're easier and cheaper to use than mice and rats, and then in conjunction with advanced AI, they can be a really good model to use for human therapeutic development for longevity and aging related disease. And so we have a couple targets in mind that we want to go after, and stay tuned on that. So.
Well, I've said, as a human who is at 100% risk for death and old age, I do look forward to that quite a bit. Thank you, Mike. Thank you, Kennedy. That's very helpful. It's so interesting to see how the Methuselah fly information can be cross-referenced, translated into the supercentenarian data, and find new hypotheses that might help all of us through this precision medicine, through looking specifically at individual or very micro population genetic targets or medicinal targets, to really improve lives in substantial ways. And I'm excited to see how this hypothesis generation, the targets you're looking at, the flies all wrap up into this. I'd love to hear a little bit more about how we're using these systems in a way, we're building these systems in a way that the data that we're using, the outcomes that we're getting are trustable, that they're accurate, that they're relevant to the biological issues that we're investigating.
I would say the fact that the Bioatomspace is something that's tangible enough to be a source for anything that can be present, let's say for anything that any AI related work that we do over the Bioatomspace. So it can be a source that a biologist can refer back to, or can do an evaluation on, let's say. So with that, I would say it's quite replicable work that you can do with what we have in the platform. So it's not something you get now and it will be gone, or you don't even know how to replicate it, but it's something you can trust on, or you can know that you can replicate it some other time if you want. Yeah. So
Yeah, just to add to that, so currently we are using publicly available knowledge sources. Of course, in our future roadmap, we plan to allow institutions to load their own private data and experiment and run our algorithms on that. But most of it currently, the experiment is being done in silico or on your computer. So we are giving the most likely prediction. So at the end of the day, the best way to ascertain it would be, of course, to take that hypothesis and train, test it on the lab. But what our system does is it shortens the time between just testing 1,000 different hypotheses. It just gives you the top 10, so you can focus on that. And this is backed by literature and publicly available resources. So this does give more confidence when doing this kind of open-ended research. But of course, at the end of the day, the hypothesis our system generates has to be ascertained and tested in a lab setting to be 100% accurate. Yeah.
Yeah, and we're hoping that it's a system that will, evolve like anything else. Our business model, as we have it outlined, allows for, a couple of different scenarios where we have sort of a a web-based, more open source version, and then, kind of a private licensed version for, your university and biotech labs that might have to keep their data private. But, we would hope that as scientists, use the platform and get their ranked hypotheses or whatever, and then they test those like actually in a wet bench setting, then they go back and they input the data that they've acquired, and then that adds to the system, right? And then it learns from that and says, okay, well, that didn't work, and then it further refines like its output for the next scientist who maybe uses it in the open source version, like maybe uses it on another side of the world or whatever, and that adds to the whole thing. And so then the system, evolves, and then maybe someday eventually you get to a point where you're not doing as many wet bench experiments, or maybe not any.
And we've recently seen that in the past year, where the requirements for animal-based animal testing has been softened. There are certain types of medicines that don't require animal or human trials. Is that correct?
Yeah, they're not technically required anymore, although everybody's still doing them. and yeah, and if you want to get investment, at least in the United States, like you better still have animal trials. So yeah. But I mean, but animal trials are still very expensive, and so this would be a tool that people could use like before they drop that, hundreds of millions of dollars or whatever on an animal trial. They would at least have a better idea that what they're about to test in an animal will at least translate and make it into a human and have a reasonable chance of actually helping a human, instead of, be like, oh, well, this, cures Alzheimer's in rats or whatever, and then doesn't do anything in a human. So
are you seeing a lot of interest, acceptance, welcoming of this approach among investors, academics, entrepreneurs, in enterprise, folks who are exploring longevity?
I'd say in longevity, where people are a bit more open-minded, yeah, people are more excited about this approach. In traditional biotech, I'd say it's still very much at arm's length. I was at JP Morgan in January, where I presented in a Tech Bio showcase, which was a co-located event to JP Morgan. And Mike was there with me. There was a few generative AI tools that people were talking about, but I was only one of like a handful of people that suggested anything beyond generative AI. Mike and I sat in on a couple of panels where there were some pretty prominent scientists actually that were talking about more cognitive architectures in AI drug discovery and research. And anytime they mentioned it, you could hear people kind of scoff in the room, like they didn't think it was like going to be a thing, or they thought it was science fiction or what have you. I don't know. But I mean, if you think of at least the way that drug development and that whole system works right now, especially in the United States, I mean, it kind of makes sense that they they would hold that stuff at arm's length, because there's a lot of things that make the drug development process in here a very long and arduous and expensive process, and there's a lot of things that make a lot of people along that road a lot of money. I mean, you have everything from, like, you have your kind of your cheap target discovery, where you have, postdocs in universities toiling away where they discover the targets, and then you have a pharma company that swoops in and licenses the targets from the universities, and then they develop that target, and then they develop it until it becomes a thing, a drug or whatever, and then they manufacture it and distribute it, and then it gets sold to the pharmacy benefit manager, and then that gets managed by health insurance companies and all of that stuff. And here comes AI. I mean, generative AI or cognitive AI, whatever you stand to use, AI, that stands to disrupt that whole system, cut discovery time in half, like cut costs in half or by more, and cut people's profits. People along that road that make all that money, of course they're of course they're gonna, their reaction to be like, oh, well, no, that's never gonna work. Like, it's not gonna, I mean, yeah. So, I mean, but at the same time, there are people who definitely see that it's not going away and that it's coming. And I mean, I've received a lot of really positive feedback from people who want to collaborate, like scientists who want to be on our beta tests, who want to use the platform, who are amassing like massive amounts of data that there aren't tools available to find patterns in that data. Particularly, people who are particularly excited are immunologists, for example. Like, the immune system is an extremely complex bodily system that touches all other systems in the body, all different kinds of cell types. I mean, extraordinarily complicated to find unifying patterns and theories of how diseases are interacting with the immune system. And so it makes sense that, like, those folks would be really interested in a platform like this. And so yeah, so that's kind of the current state of things. I mean, it's kind of one of the things that, like, traditional biotech is sort of leery of, but they also they can't ignore it. And I mean, it's coming. So
yeah, so many industries are experiencing disruption right now. Bio's not an exception. Some component of each of those industries is drawn kicking and screaming, and the rest are leaping ahead to use new tools and new ideas, even the generative AI components, to do so. And I can see this is no exception. It's interesting to see how LLMs and neural networks, the advances there, have driven that acceptance or understanding that this is coming, and it is transformative across industries. Thank you. And then I'd love to touch for a few minutes with the team on the future outlook. What should we expect in 2025 and beyond for Rejuve.bio as a platform, as a business, and what should we be looking for?
Well, I think we're hoping to get a demo out, like a beta version that's functional enough for biologists to actually do some real research on, by the summer or the fall at the latest. And, we have people lined up, interested in testing it. And so, yeah, we're hoping, hoping at that point we'll be able to have up the virtuous development cycle where we can get rapid feedback to inform our ongoing development, both for our own platform and for Hyperon as a whole, as that develops, and that and as we integrate its developments into our platform, that we can both improve that and improve the larger Hyperon development goal towards developing true AGI.
Yeah, as Mike said, yeah, we do have a pretty good list of beta testers lined up, both in small biotechs and academic research labs, to give us good feedback on a beta version, and so then hopefully like an official launch like sometime next year. But yeah, so that's where we are, and then hopefully that can help democratize this type of research and start helping make a drug discovery better.
We're also planning on finally publishing some of the Methuselah fly data and what our findings are by the summer to go along with the beta release.
It's such an interesting multi-pronged company to be both releasing this super advanced AI as well as longevity insights themselves. So it's amazing to be seeing this project. It's been internal for so long. Our researchers have been looking hard in this direction, developing hypotheses and AI that can help with bio and how to, even just extract biodata, some best practices there, extract biodata into a knowledge graph, and then explore it as Hedra showed us, and generate hypotheses as Abdul showed us. And now to be on the edge, the brink, the year that we're going to take that out into a wider audience and find out what others can do with these great tools, with this great information, with all the progress that we've made that now gets to be launched into the wide world and really improve it in a beneficial way. It's exciting. I'm excited. This has been an excellent conversation. I think that there's so much more we could talk about. I'm personally very curious for next time to talk about how we're handling some of the scalability components. There is so much biodata out there, and it's increasing and growing every day. But another time and the future to look at some of these launches, the improvements, the user base, the growth, and perhaps how some of these hypotheses and understanding that we're growing about the Methuselah flies can have real impacts. I look forward to seeing folks here, the Rejuve.bio people, at the BGI event in May in Istanbul. I hope everybody can join us there, either live or streaming the virtual event. This is such an important component for SingularityNET, our passion on building beneficial AI, beneficial AGI, AI that's helping solve real-world problems for real people, personalized down to precision medicine for individual genomics in the future, and not just making money for a company, but actually providing benefit, creating a world that we all want to share and live in. Thank you all for joining here today. I hope everyone enjoyed it. I had a great time, and we look forward to talking with you all again soon. Thanks, Haley.
Thank you, Haley.
Thank you. Thank you.